Hello. In today's video, we're going to be talking about the cell. In the introduction, we talk about how the cell is the structural and functional unit of life. It's the smallest structure that's capable of performing all the functions of life. It's so small that you need to use a microscope to see it. You have a little diagram in here showing you the size of a person, and the average cell, keep in mind every unit increases by 10, power of 10. So the cell, we are talking about micrometers, millions of a meter. Bacteria, they're about the size of the organelles. They're very, very small. Let's just put it like that. I don't need you to memorize sizes. They're so small, you need to look at them with a microscope. And in general, the size is limited because of the volume increasing much quicker than the surface area. Have you ever wondered why, instead of having several trillion cells, why don't we just have a few thousand big cells that can do the same job? That is the problem. Let's assume, for simplicity, that the cell is like a cube, okay? Just because I can do math with this. You know it's not a cube, but it's a good approximation. Okay, so let's assume that the size of the side is going to be x, okay? The surface of the cell, you know that each square would be x squared, and you have 6 sides, so it's 6x squared. The volume, on the other hand, for this one is going to be x cubed. Now assume the cell doubles in size. Okay, so this is going to be 6 times 2 squared is going to be 4x squared, so it's 24x squared, and this is going to be 9x cubed. Okay, so basically the idea is that the volume increases faster than the surface, and the volume of the cell tells you how many organelles you have, how many chemical reactions. You need oxygen, you need nutrients to get into the cell, you need carbon dioxide coming out, you need waste products to come out. So you need to have enough surface to maintain the cell alive. At a certain point, the cell is too big, there's not enough surface, and it's not viable. That's basically what this statement here is trying to say. Cells are very variable. I'm giving you some examples in there. These are from the nasal sinuses. They're animal cells. You can see that they're kind of rounded. This one's in here. They're onion cells, onion skin. These are plant cells, and you can see that they have kind of straight walls and all of that. Very different in size too. And here you have some bacteria that are seen with an electron microscope because of how small they are. So the cells are from different organisms that are very different, but they're going to have certain characteristics that all cells must have. So basically, all cells are surrounded by a plasma membrane. That's what wraps the whole cell. It's a boundary to the surrounding environment. So the major function of the cell membrane is to control what comes into the cell and comes out of the cell. We're going to have a whole chapter on that, so don't worry about it yet. Inside the cell, you have the cytoplasm. And in the cytoplasm, you're going to have organelles. We'll talk about that in a moment. We can classify cells into 2 major groups. Prokaryotic cells, they don't have anything inside that's surrounded by a membrane. Okay? We are going to see the different organelles. They do have ribosomes because they make protein, but ribosomes are not surrounded by a membrane. All of these organelles you can see in there, they all are surrounded by a membrane, so you don't find them, including the nucleus. Okay? So most people just said prokaryotic cells don't have a nucleus. Well, they don't have membranes surrounding nucleus. If they do have organelles surrounded by nucleus, they're called eukaryotic, and all those little structures are called organelles, and we are going to be looking at the common organelles and what do they do, how do they look like. Let's real quickly talk a little bit about prokaryotic cells. like a nucleus, again, a membrane surrounding nucleus, they're going to have nucleic acids, okay? They're going to be located in a special area of the cell, but there's not a nucleus separating the nucleic acids from everything else. Most prokaryotic cells are unicellular, meaning they're made up of only one cell, but they tend to group. So you can have cocci, those are isolated bacteria. Streptococci, they are strings. Each is an individual cell, but they don't form an organism. They just live like a colony. Okay, they all live together, but each cell is individual and has to perform all of the functions to live. Or they can just cluster, like in Staphylococcus. Most prokaryotic are very good to adapt to almost an environment. I'm not saying each one, but you're going to find prokaryotic cells in just about any environment. Different ones, yes, but you can find them everywhere. And this included bacteria and the archaea that we talked about when we was talking about taxonomy. Main characteristics of prokaryotic cells, you have a cell wall outside the plasma membrane. Don't worry about what it's made of. And actually, depending on how much amount of peptidoglycan, you can have more than one kind of cell walls, and many antibiotics work on preventing bacteria to making the cell wall, which makes them, they cannot reproduce, they're more susceptible to damage, things like that. And of course, depending on the amount of peptidoglycan that they have, you're going to have to use different antibiotics. But most bacteria have a cell wall. Many, but not all, have a capsule. That's another layer outside that is surrounding the plasma membrane. Some people say it's a little bit sticky, so it helps bacteria stick to places. That goes right there with the fimbria, those short appendages that are coming out, that helps attach to the surface. So why is it so bad, for instance, when you are sick, to sneeze on your hand? Well, and I realize colds are caused by viruses, but let's say you have a strep throat. That's a bacteria. So you cough, and you've been taught to always cover your mouth with your hand. You cough, you get the bacteria in your hand. They stick to your hand. The combination of the capsule and the fimbria cause that bacteria to stick to your hand. Then you're going to be leaving the room and you grab the door handle. You leave bacteria in there. The next person touches the door handle, gets the bacteria in the hand. And the next thing, you're rubbing your eyes, touching your nose, and you see how easy it is for bacteria to spread. Combination, like I said, the capsule, that's sticky, but the fimbria, that's like a Velcro, helping stick. Many bacteria, not all, but many have a flagellum, a very long appendage for movement. There's a few prokaryotic cells that have more than one, but some instead are going to have short cilia. They're like a flagellum, but they're short, and many of them, and it's almost like oars on a boat, and the bacteria can row. The nucleoid is the region where the DNA is present. Again, there's no membrane surrounding it, but there's a region where you're going to find the DNA. That's called the nucleoid. And by the way, they didn't say you in here, but something interesting about at least bacteria is that the DNA is always circular. Okay, and like eukaryotic DNA. And they do have ribosomes, we said. You need them for protein synthesis, but because they're not surrounded by a membrane, you can find them in prokaryotic cells. Eukaryotic cells, on the other hand, are going to be bigger. They have a eukaryotic cell in pink. They have a bacteria in green, to give you an idea on the difference in size. They do have a nucleus surrounded by membrane and organelles surrounded by membrane. Okay? Organelle means a little organ. And we are going to study all of them. And eukaryotic cells include animals, plants, fungi, that's mushrooms, yeast, and all that, and proist. So there you have an animal and a plant cell, and don't worry about all of these names. I was tempted to erase them. We are going to look at them, okay? And we are going to look at each one, what they do. Right now, I just want you to see the difference between the animal and the plant cell. The animal cell is kind of rounded. Now, it can be spherical, it can be flat, something like that, but you don't see no many straight lines. Look at the plant cell. It has kind of like straight wall. And that's a perfect term because it is the cell wall that causes the shape of the plant. And the cell wall protects it, maintains the shape. We are going to see when we study about osmosis, things like that, the function of the cell wall. But at this point, it keeps the shape of the cell. Most organelles are going to be common. There's a couple of them. The lysosome, for instance, is commonly found in animal cells because animal cells are more likely to swallow something to digest it. And the lysosome we're going to study is at the garbage disposal. All of the waste leftovers going to be destroyed in there. The plant cell may have lysosomes, but it's less common. It still needs to remove all organelles or something like that, but the cell is not quote unquote eating anything, so that makes a difference. Notice that the plant cell has a really big vacuole. It's filled with liquid and all that. It helps keep the shape of the cell. The animal cell may have some vesicles, but you don't have a big vacuole in the center. Okay. Sorry, I was trying to, can hardly read in here, the vacuole, sorry. That's what I was marking. And the other thing that I want to mention is the chloroplast. That's an organelle that only appears in plants because that's where photosynthesis takes place. Animal cells don't do photosynthesis, so they don't need. So understanding that there's some organelles that are more common, like the lysosomes are more common in the animals, or that they're present only in a particular type of cell, like chloroplasts are only present in the plant cells, I like to just go over the structure of the average cell. Okay? We begin with the plasma membrane at the outside. It's a phospholipid bilayer. Remember when we studied phospholipids, we said it has a polar end and then two non-polar tails. It's represented in there by having two layers. That's what the bilayer means. You can have the polar heads towards the outside of the membrane, the non-polar in the inside, and then of course on the other side you're going to have polar heads again. And we mentioned in that video that the reason for this is because where you're looking outside of the cell or inside of the cell, there's going to be water. And polar ends are going to form hydrogen bonds with the water. They mix with water real good. The non-polar, the hydrophobic tails don't like water, so they kind of hide in there. There are other components too. We're going to be talking about, sorry, I've missed the proteins. They're going to be embedded. They're going to have a bunch of functions. We'll study them later. It's another video. There's some cholesterol. Cholesterol controls the degree of fluidity. These proteins are going to be moving. So you need the phospholipids to be fluid enough to allow for the proteins to move, but not that fluid that it melts. So cholesterol is going to be in charge of controlling fluidity. And you have carbohydrates. You see some carbohydrates sticking out. They're usually going to be used as ID tags, saying, I am a liver cell, I belong to Dr. B. That's why when you get a transplant, yes, it is a liver cell, but if I get a liver cell that comes from my husband, my body is going to look at the carbohydrate and say, you don't belong here. And it's going to attack it. That's why people who have transplants have to take anti-rejection drugs for the rest of their life. The main function still is going to be regulating the passage of substances. Okay? It's going to prevent things that the cell doesn't want to get inside. It's going to allow others or even bring them in actively. And it's going to also remove things from the cell. Some cells, especially those that are specialized in absorption, that the ones that line your small intestine, they're going to increase their surface by having these finger-like projections on the membrane that are called microvilli. Basically, let me put it this way. You're going on a trip and you're packing clothes and you have a size of a bag of a certain size. What if your clothes are too big? What do? You fold them. Because when you fold the clothes, you can pack more clothes in smaller, right? Let's say that I only have room between here and here for a cell. Okay, I have this distance of plasma membrane, but the same distance, if the cell starts having projections, which one has the longest red line? The one that has the microvilli. You increase the surface. And like I said, cells like the ones that lie in your digestive system that are going to be absorbing, the more surface, the better they can do their job. We said the cytoplasm is going to be the inside, everything between the plasma membrane and the nucleus. It has organelles that are suspended in a substance that's like a gel. It's called the cytosol. So cytoplasm is everything inside the cell, except for the nucleus. Cytosol is the gel-like substance, where the nucleus, the cytoskeleton, we're going to be talking about that, and a lot of water is in there. Did you know that cells have skeletons? I bet you didn't. It's not really a skeleton like you're thinking. It's not made up of bones, obviously. But that's what cytoskeleton means, skeleton of the cell. There's three major types of fibers: microfilaments, intermediate filaments, microtubules. We're not going to get here into what each one is made of or something like that. If you're interested in this, I recommend you take anatomy and physiology. But all of them together, they're going to maintain the cell shape. Notice the intermediate filaments are crisscrossing the cell. The same thing, the microfilaments are right under the plasma membrane. So together, they maintain the shape of the cell. They assist in the movement of the cell because when you have cilia or flagella, and you can have them in eukaryotic cells too, they're made up of microtubules. And that allows the cell to move, or to move the environment around them too. And organelles can move. Sometimes they need to move. So once again, you have filaments that cross the cell almost like railroad tracks. And certain organelles can be moved from one place of the cell to another following those tracks made up by the cytoskeleton. Most of the organelles that we're going to be looking at are part of what we call the endomembrane system, meaning a system of membranes inside. That's what the endo means, inside of the cell. And it's basically how materials are made in the cell and how they're transported. So at this point, we're not going to get into each one. Let's say that everything begins with the nucleus. Then the information is moved to the endoplasmic reticulum. That's going to make some products. We'll go into all of that. It's going to be packaged in the Golgi apparatus, and then they form either vesicles or lysosomes that are going to go to plasma membrane, and they're secreted. So the information flows from the nucleus to the endoplasmic reticulum to the Golgi, and we're going to study each one slowly. We begin with the nucleus. It stores genetic material, DNA. Every cell of an individual contains exactly the same DNA. Remember, you began as one cell that divides over and over and over, and the DNA directs the characteristics of a particular cell. But you don't have pure DNA. What you have is DNA associated with some proteins that make it more stable and helps control expression of genes and all of that. So that mixture of DNA and proteins is called chromatin. And chromatin will be very, very long, but it coils. It coils, condenses, and sometimes it relaxes, depending on the stage of the cell cycle. We'll see all of that. But when it's condensed, we'll call it a chromosome. So looking at the nucleus, the inside is the nucleoplasm. Just like the cytoplasm was the inside of the cell, the nucleoplasm is going to be the inside of the nucleus. In the center, you can see that there's something darker, it's the nucleolus, and there's a particular type of RNA, ribosomal RNA or rRNA, that's made in there. This is going to be very, very important because it forms the ribosomes. Ribosomes are made up of rRNA and protein, and ribosomes make protein, so they're essential for the cell. You have a double membrane surrounding the nucleus. It's called the nuclear envelope. Like I say, it's a double membrane, but it has openings, nuclear pores that can open and close to allow certain things to get in or come out. Like, for instance, you make the rRNA from the nucleolus, but to make ribosomes, it has to come out. So it comes out to one of the pores. So now the rRNA is in the cytoplasm and can bind to some proteins and make a ribosome. Same thing we're going to see when we look transcription translation. Messenger RNA is going to be coming out, and these pores can be controlled by the cell. So they will allow certain things to come out, but keep other things inside. It's a protection for the DNA. We talk about the endoplasmic reticulum. The information comes from the nucleus to the endoplasmic reticulum. And there's actually two types, the rough endoplasmic and the smooth endoplasmic reticulum. This name comes from the first time people saw it under the electron microscope. Some look very smooth and some was bumpy. They call the bumpy rough endoplasmic reticulum because it's covered with ribosomes. Okay? So in the rough air, you're going to make, modify, and fold proteins. It's covered with ribosomes. With the ribosomes make proteins. So all the modifications the protein needs to undergo from the primary structure to secondary, tertiary, maybe even quaternary structure, they take place in the rough endoplasmic reticulum. The smooth looks smooth because it doesn't have ribosomes. Instead, it's going to synthesize carbohydrates, phospholipids, steroids, and depending on the cell type, sometimes it has specific functions. We're not going to get into that right now. Okay? You have a little diagram in here. Obviously, you can tell the difference between the rough and the smooth. If in an exam you're supposed to identify the endoplasmic reticulum and you really cannot tell the dots, it happens sometimes. Okay? One thing that you want to know is that the rough endoplasmic reticulum is flat, like flat sacs. Okay? While the smooth endoplasmic reticulum is more like open tubules. So even if your picture is not good enough, you can still look at the overall shape and tell which endoplasmic reticulum you're dealing with. Notice the continuity between the nucleus, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, and then let's say something about the ribosomes real quickly. The site of protein synthesis, we say that they're form of ribosomal RNA and proteins. And we already talked about they're attached to the rough endoplasmic reticulum, but many are free-flowing in the cytoplasm. Basically, and that may not be 100% accurate all the time, but if the protein is going to be kept in the cell and used immediately, there's no need to package it, so these loose ribosomes can do it. But if this protein is going to be packaged either to store it or to send it outside of the cell, then it's made in the rough endoplasmic reticulum because it's going to be continuous with the Golgi apparatus. Okay? So you can see in here how after the endoplasmic reticulum, you have the Golgi. It's a bunch of and stack of curved sacs in there. And some people call that the shipping center of the cell because it packages. The proteins that were made in the endoplasmic reticulum, they're packaged. Like I said, some are going to be stored, they need to be protected, some are going to be shipped outside of the cell. It will do some minor modifications to the proteins, but the main thing is sort them by what are you going to do with them, if necessary, their packaging, and distributes them. So picks a protein and says, oh, we're going to keep this one, we're going to use it right now, I'll just send it to the cytoplasm. Oh, this one needs to go out of the cell. So it's going to be packaged and can go to the cell where the two membranes fuse and that protein comes out of the cell. How does the Golgi apparatus know? Because the proteins have some tags attached to them that work like a zip code. Think on the Golgi apparatus of the post office. When it reads the zip code, it says, oh, this protein has to go here, this one's going to go there, this one's going to go to that place, by reading the zip code in there. So whatever is made is going to be used somewhere inside the cell, or it's going to be excreted outside. Lysosomes, I don't have a drawing because it's nothing more than the vesicle enclosed by a membrane, but it contains digestive enzymes. So some people call that the garbage disposal of the cell. It breaks down unwanted and foreign substances, worn out parts of cells. If you use a white blood cell for instance, a lymphocyte, and there's a bacteria here. What do? You swallow the bacteria, right? But what would we do if the bacteria is alive? It reminds me of the first antivirus I had on my computer. It was a free version you could download, and every so often I get a message. A virus has been detected and is quarantined. I didn't like it. How do I know it doesn't get out of quarantine? Finally, I got a message one day saying that they have quarantined a Trojan, and that's when I said, I need to pay for the antivirus. And I actually bought a good antivirus that whenever it finds something, it destroys it. I don't want it to be quarantined in there waiting one day to escape and burn my computer. Same thing, this bacteria could escape. But if you have a lysosome, the bacteria gets enclosed into the lysosome digests it, the bacteria is dead, gone. It cannot escape. Vesicles and vacuoles are pretty much the same thing, membrane-bound sacs, but their function is going to be storage and transport. The only difference between the two of them really is the size. The vacuoles are bigger, and vesicles tend to be smaller, and vesicles fuse. We are going to be talking with our membranes. But we talk about the central vacuole in a plant cell is filled with water. Now it's going to have pigments. In some cases, it may have toxins, it may have some other substances, but it provides support to that cell by putting pressure on the cytoplasm. Vesicles tend to be smaller and diffuse with others, like we was talking about the cell that swallow the bacteria. Well, when you swallow bacteria, it's surrounded by a membrane. Let me try again. So you have your cell and you have the bacteria. So the cell is going to start trying to surround it, and when these two ends touch, you end up with the cell inside, with the bacteria inside of a vesicle. Now the lysosome comes in here, and when they fuse together, then it digests the bacteria. This is just one example where a vesicle fuses with other membranes for a particular function. Chloroplast, that's the place where photosynthesis takes place. That's when all solar energy is used to make carbohydrates in plants. Algae will do that too. Actually, some people claim algae today produce just as much or more oxygen than trees. The structure is very typical. You have a double membrane. The inside, filled with fluid, is called the stroma, the inside of the double membranes. Then you have thylakoids that are sacs. They look like a pancake, okay? Because they are very flat, and a stack of pancakes, a stack of thylakoids, is called a grana. And there's several of them. And photosynthesis requires chlorophyll. Everybody knows the chlorophyll is located in the thylakoid membranes. When we study photosynthesis, all of these terms will become very important. The mitochondria, you're going to find it in all cells. That includes also the plants and the algae. Just like chloroplasts appear only in plants and algae, mitochondria appears in animals, everywhere, okay? Because they're the site for cellular respiration. That's where carbohydrates are breaking down with oxygen to produce ATP, that has the energy for the cell, okay? Just in a way like the chloroplast is bound by a double membrane. They have a fancy name, inner membrane and outer membrane. There's a matrix inside, filled fluid. You can see the similarity between that and the chloroplast. And the foldings of the inner membrane are called cristae. And every time you have a membrane that folds, what do we say it is? So you can have more membrane. And this is very, very important to have more membrane, because this is where the last step of cellular respiration, the actual synthesis of ATP, takes place. So the more membrane, the more ATP the mitochondria can make. And something interesting, they contain their own DNA. It's going to be very similar to bacterial DNA. It's going to be circular too. And when we talk about evolution, you see that some people believe that mitochondria were actually bacteria that were swallowed by another cell, and it was useful for it to stay there. My question is always, how did the cell live before forming the first mitochondria, which of course there's no answer for it. Another organelle, that's another vesicle, although this time is only single membrane, is the peroxisome. It has a number of functions. It oxidizes fatty acids. Fatty acid metabolism is a little bit complicated, and amino acids. It detoxifies. It detoxifies poisons. It detoxifies alcohol. Have you ever wondered why it's bad to drink too much? And I'm not talking about getting drunk one day. I'm talking about people who drink consistently. You may be a social drinker, but if you do it consistently, what happens eventually? Cirrhosis of the liver, because the detoxification of alcohol produces some toxic molecules. Don't last very long, but you do it over and over and over, and it obviously affects the cells. But the main function of peroxisomes, and that's where it gets the name, is to break down the hydrogen peroxide that is formed in oxidation reactions. What's wrong with hydrogen peroxide? It's H2O2. It's toxic to cells. That's why if you put it on a cut, it kills bacteria. But we don't use it a whole lot because we found out that it actually damages your skin too. If you have it inside the cell, it will kill the cell. But the peroxisome breaks down the hydrogen peroxide into water and oxygen. These are the bubbles that you see. Hydrogen peroxide in the bowl doesn't have bubbles. But the moment you put it into the broken skin, it starts bubbling because you have an enzyme that breaks down the hydrogen peroxide. So the enzymes in the peroxisome break down the hydrogen peroxide that's been formed in the metabolism. You can't help it, but it breaks it down to oxygen and water that will not harm the cell. Outside of the cell, we call that the extracellular matrix, and it's a bunch of stuff secreted by the cells, mostly glycoproteins. That is a mixture of a sugar with a protein and a particular protein called collagen. The idea is like a glue that holds the cells together. That's what holds the cells together in the tissue. They're not holding hands. They don't separate. They don't break apart. And also it allows the cells to communicate with each other. Okay? We're not going to get into too much detail, but please don't worry. There's 4 types of connections between cells that are called intercellular junctions, depending obviously on the need. Okay? So you have plasmodesmas. You find that in plant cells, because we are going to see that fluid that carries water or that carries sugar has to be moved throughout the plant. So you have an actually big gap where these 2 cells come together for water to flow through it, carrying many times nutrients in there. So plasmodesma is a channel between plant cells. Now, tight junctions, you find them in animal cells, and they're watertight. They're like a zipper. Okay? If you're wearing a windbreaker, it starts raining, if you zip it up, you're not going to get wet, right? So whenever you have tissues that are going to subject to stress, you have tight junctions to make sure that the cells don't separate. Think, for instance, the cells that line your stomach. The digestive juices inside the stomach and the hydrochloric acid would be very, very dangerous. If they could seep slowly between the cells, you could get out of the stomach and cause damage. But because you have tight junctions, they're watertight, they are in the stomach, they cannot come out. Desmosomes are another kind of joint for animal cells, but basically what you have is a number of fibers, mostly proteins, but not only proteins, that hold them together, and you don't have the whole, using the tight junction, you have a whole line that's like seal. It's like a ziplock in there. Desmosomes, you have a little area, and then you have some area that there's nothing, and then you have another desmosome up here, another desmosome down there. So it's almost like Velcros. You have little patches that hold the cells together. The 2 walls are not touching each other, so the room water can get in there, and there's areas that are not held together. Finally, gap junctions, they appear also in animal cells whenever you need a channel. You see, you have this channel surrounded by proteins, but the way this flower-like structure is made up of several subunits, and they can open and close the channel. But when you need very quick communication between two animal cells, like think on the muscle in your heart, every cardiomyocyte needs to contract at the same time, or else the heart will not work. If everybody would contract whenever they feel like it, well, that's what fibrillation means, and the heart cannot pump. So it's kind of like the cells say, okay, on the count of three, everybody contracts. 1, 2, 3, contract. 1, 2, 3, contract. But of course, they don't count to 3. They don't say contract. They move ions through there so they can communicate very, very quickly, so they all contract on time. Hopefully this is not very complicated, but if you want to work with me one-on-one, if you want to look at any of the charts I have made, summarizing everything, find the link to my recent Turing profile in the video description, make an appointment, and I'll be glad to help you.